Alright, here we are almost a month later in the Penicillium growth experiment. On July 24th, I took a lemon sliced in half, just like this, and some water, put it in a Tupperware container, covered it, and put it in the back of a cabinet in the dark. And I checked for Penicillium mold every couple of days. And after about a week and a half, the telltale olive green Penicillium showed up and was growing pretty well. Definitely had a powdery look to it, and when I looked at it under the microscope, you could see a bunch of spores and the fan-shaped finger-like conidia. But that has seemed to have been replaced by this white fuzzy mold. It's fluffy in its own right, but not powdery like Penicillium, and it doesn't look like there's any actually spores. This looks like vegetative mycelium. So there's no reproductive spores or reproductive parts on it, and I looked at it under the microscope as well, and it just looked like white stringy mycelium. So this is either a different species, or it's maybe penicillin or Penicillium that's turned into a different stage in its growth, because I think I've seen some white Penicillium species. But anyway, to try and liquid culture it, what I'm going to do is take another lemon, slice it into pieces, boil it in enough water, and take some samples off of this, put it into each of these, and see if I can get a liquid culture. Because the last time I tried to distill D-limonene from lemons, I ended up leaving this in the round bottom flask because it was so hard to take out for a couple days, and I came back and there was a huge mat of mold growing on it, which I assume was the penicillin. And when I was heating it, trying to do vapor distillation, broke down all the sugars and nutrients, so it was easier for the fungi to digest and grow. So I'm going to try and do the same thing here. Alright, so step one is to put the parent culture aside, which we're going to try and isolate the strain that's growing from. I have a pot here, 2 glasses that I'm going to use as inoculation flasks basically, because I don't have any glassware. So I pre-measure just about half in each cup. I'm going to pour that in there, turn on the heat to high, and then I'm going to throw in all these lemon pieces. And the penicillium that grows specifically on citrus fruit have obviously adapted to growing in acidic conditions and living off whatever nutrients are in the lemons. So by heating them and breaking them down, you make the nutrients more readily available, and hopefully the thought is that the fungi mold will grow a little bit better, and in maybe a couple days we'll have semi-clean or semi-pure cultures of whatever it is that's growing on here, and then we can extract some DNA and do ITS amplification with PCR and barcode the fungi. So this is going to take a while to boil, so I'll come back when that's done. Alright, so I've boiled the lemon peels in water. I ended up using a potato masher and kind of breaking them down. It's still a little hot right now, 72 Celsius. And I'm going to add one more container to the experiment. I'm going to do one container that is the inside of the lemon, which might have some of the old spores of the olive green penicillium. I took a little core sample of the lemon, put it on the microscope, and it did look like there was some spores down inside in the area that was still olive green. I'm going to do another container with just the white fluffy mycelium on the surface of the lemon, and then I'm going to do one more container, which I'm just going to dump all of these lemon rinds and some of the liquid into this container and not inoculate it with anything, and see if anything that had been living on the lemon peel originally will grow in the liquid, sort of like a control. And these other two I'm going to filter out the bigger chunks and just grow them on the liquid. So once this cools down, a little bit more. I'll put it into the plastic.